Hello everyone, welcome to the ITLS Academy. Today we are going to discuss another topic in the series of food preservation, and the topic that we are going to discuss is about the antimicrobial enzymes. Okay, we are going to discuss that how this antimicrobial enzyme can preserve the food product, can preserve the food products. Okay, now, and how antimicrobial affect the shelf life of the food product, that we are already discussed about it, and the video related to this are already uploaded in our account. You can visit our YouTube channel and see that. But here we have to see that what is the antimicrobial enzyme effects on the shelf life of the food product, on the shelf life of the food products, how it can increase, or you can say that how can we extend the shelf life of the food product. Along with that, we also discuss about the mechanism of the antimicrobial enzyme, and then we discuss about that how what is the combining effect of this antimicrobial enzyme with the other preservative technique, and what is the application of this antimicrobial enzyme. Basically, we have to see that how these enzyme can affect the microorganism present into the food product, and how can affect the chemical and the enzymatic reaction occur into the food product. Okay, ultimately the end point is that how the shelf life of the food product are extended. So we have to focus on this. So let's start the lecture. Antimicrobial enzymes, what they are the natural biocatalyst. If we talk about what is this antimicrobial enzymes are, so it is nothing, it is a natural biocatalyst which is used to inhibit the microorganisms. Okay, inhibit the growth of the microorganisms, or you can say that the destroying the microorganisms. Now this microorganism uh, affect the quality, or affect the quality of the food product, affect the quality of the food products. Okay, so we have to discuss that how these natural biocatalysts can inhibit that microorganism so that it cannot affect the shelf life of the food product. These antimicrobial enzymes are present in plants, animals, and microbes. They form a key part of the natural defense systems. Importance of the antimicrobial enzymes. So these antimicrobial enzymes provide the protection against the microbes. Now these microbes are of two type. The one is your spoilage microorganism, and another one is your pathogenic microorganism. These pathogenic microorganisms have a capability to cause disease into the human being. They have a capability to cause disease into the human being, and these spoilage microorganisms spoil the food product and release the toxin into the food product. Now this toxin can have a harmful effect on the health of the human being. So we have to discuss about it. Okay, now these antimicrobial enzymes provide the natural protection, and this protection are provide against the harmful microbes. Okay, it also reduces our dependency on the different antibiotics and the chemical preservatives, biodegradable and environmentally safe. So these antimicrobial enzymes, they are what? They are biodegradable and they are environmentally safe, means that it cannot be affect the environment. It does not cause the pollution into the environment. Now, classification of the antimicrobial enzyme. So it can be classified by two ways. The first one is your on the basis of their action, and the second part is that on the basis of their origin. If we talk about the first classification, which is based upon the action of the antimicrobial enzyme, so it can be classified as a cell wall degrading, membrane disrupting, nucleic acid degrading. So it can be classified into the three part, that is your cell wall degrading, membrane disrupting, nucleic acid degrading. Okay, now the second classification is based upon the origin. So on the basis of the origin, it can be classified into the three part, that is your animal, on the basis origin from animal, plant and microbial, means that these animal, plant and the microorganism are the source of the antimicrobial enzyme. Antimicrobial enzyme. Okay, means that we get that antimicrobial enzyme either from the animal, either from the plant, or either from the microorganisms. Okay, so we are going to discuss one by one. Now, the lysozyme is the first model of the antimicrobial enzyme. Lysozyme is what? It is an antimicrobial enzyme, and it is the first model of the antimicrobial enzyme, and it is found in ears, saliva, egg white. Lysozyme are found in ear, saliva, and egg white, and it, it can break the linkage present into the bacterial cell wall. And which linkage? That is your beta 1,4 linkage. Okay, these beta 1,4 linkage present into the bacterial cell which join the bacterial cell. Okay, so these lysozyme attack on these linkage, that is your beta 1,4 linkage, and the bacterial cell wall destroy it. Effect against the gram positive bacteria. Okay, so a gram positive bacteria means that when we do a gram staining, it give a crystal violet color. It give a crystal violet color when we perform that gram staining. Okay, now the next is your mechanism action of this lysozyme. Okay, so these lysozyme bind to the peptidoglycan. Okay, see, peptidoglycan are present in a as a thick layer in case of the gram positive bacteria. That's why the lysozyme have more action on the gram positive bacteria. Okay, lysozyme bind that peptidoglycan layer which are present as a thick layer in that gram positive bacteria. This lysozyme bind with that layer. Okay, then it cleaves the glycosidic bonds and weakens the cell wall, causing the lysis. Now what happened? Then it cleave the glycosidic bond present into that bacterial cell, due to which the cell wall of the bacteria are weak and ultimately it lysis, means that it break down. Next is your phospholipases. Phospholipases is another type of the antimicrobial enzyme. Antimicrobial enzyme which hydrolyze the phospholipids. which is present into the microbial membrane. Okay, these phospholipid present on where? These phospholipids are present into the microbial membrane, and when phospholipases are applied, so this phospholipases hydrolyze the phospholipids, and when the phospholipids are hydrolyzed, it disrupts the cell membrane integrity, due to which the death of the microorganism occur. Next is your nucleases, that is your DNSs or RNSs. Okay, so nucleases break down the microbial DNA or RNA. Means that the microbial DNA, like double helix structure and the single helix structure, is break down by the microorganism, that is by break down by the nucleases enzyme, and it also prevent the formation of the biofilm. When the nucleus enzyme attack and the microbial DNA or RNA breakdown, then the formation of the biofilm will not occur. Next is your proteases. Proteases degrade the microbial proteins. Okay, so we all know that microorganisms consist of the protein. Microorganisms consist of proteins, and the toxin also present into it. Now when we uses a protease enzyme, what they do, they degrade that protein present into the microorganisms, and also affect the toxin present into the microorganisms, due to which the degradation of the microorganism occur, or you can say that the death of the microorganism occur. Now there are two examples of the proteases enzyme, that is your trypsin and pepsin. They are what? They are the proteases enzyme which affect the microbial protein and the toxin. Now the next is your antimicrobial peptidoglycan hydrolysis. So it can be derived from the bacteriophages, that is your endolysin, and it the target of this antimicrobial peptidoglycan hydrolysis is what? The bacterial cell wall, and it can be derived from the bacteriophages. Plant derived antimicrobial enzyme. Now the first part is completed, that is your animal derived antimicrobial enzyme. Now the second is your plant derived antimicrobial enzyme. So the first one is your chitinase target fungal cell wall. So a chitinase target fungal cell wall, they what they do, they hydrolyze the beta one third glucanases, and this beta one third glucanase is converted into the glucans. Okay, when it is converted into that glucans, then a difference act occur with enzyme like activity. That act is very similar to the enzyme like activity, and it is occur by the chitinase target fungal cell wall. Now the next is your microbial derived antimicrobial enzyme. So it can be produced by the microorganism, but here see, microorganism are of two type. The one is your harmful, and the second one is your beneficial. Again, this harmful is of two type. One is your pathogens, and the second one is your spoilage. So we are not talking about the harmful microorganism, because this harmful microorganism cannot use in the production of the antimicrobial enzyme, but this beneficial microorganism have a role in the production of that antimicrobial enzyme. Okay, this outcompete the pathogen. So these beneficial microorganisms have a competition with the pathogenic microorganisms. For example, bacteriocin, which are associated enzyme. Bacteriocin is what? It is a associated enzyme. So this is about the microbial derived antimicrobial enzyme. Now the next is your Application in food preservation. So these lysozyme used in cheese industry for the prevention of the Clostridium. Clostridium is what? Clostridium is a bacteria and it is very harmful bacteria. But when we use a lysozyme in the production of the cheese, then it can uh, prevent the cheese from the Clostridium. Chitin is present in a fruits which protect the fruit from the fungi. So here lysozyme used for the production from the Clostridium and chitin is used from the protection of that fungi. Application in medicine. It is used to treat the different infection which is caused by the bacterial. Like it can degrade the biofilm on medical devices. Application in agriculture. Plant spray with the enzyme, they are called are genetically modified plants. Okay, because it is expressing the antimicrobial enzymes activity. Suppose that we have a plant. Now what we have to do, we spray that plant with the help of the enzyme. And which type of the enzyme? Which is antimicrobial enzyme, means that it can protect the plant from the microbial attack. Now when we do this, then that plant are converted into a genetically modified plant and it is expressed as a antimicrobial enzymes. Enzyme as a alternative to the antibiotics. Antibiotics is used for the treatment of the bacteria, but we can use enzyme as a alternate of that antibiotic. Now there have a specific action against the pathogenic microorganism, means that the microorganism which have a capability to cause disease into the human being, not microorganism, especially we talk about the bacteria which have a capability to cause disease into human being, lower the risk of resistance development. Combining the enzyme, this antimicrobial enzyme with the other preservation methods, the first concept is of the hurdle concept. Okay, hurdle technology means that we mix up the different technology of the food preservation and then that technology affect the microorganism and increase the shelf life of the food product. Mild methods combine for strong microbial control. Benefit of that combining enzyme with other method, like it can reduces the required road dose of each holder, broadens antimicrobial spectrum, and it also minimizes the sensory changes. Okay, when we combine these antimicrobial enzyme with the other method of the food preservation, then what they do, we required a very less dose of the each hurdle technology. Okay, and we get what we get, a broad antimicrobial spectrum, and along with it also minimizes the sensory changes which occur into the food product, which is very important, because if the sensory changes occur, then that food are not in a good quality, and if we consume it, so then obviously there is some effect on our body, and we don't get that taste that we get from the original one. So this can minimizes the sensory changes occur into the food product. Mild heal treatment plus the enzyme. Now suppose that if we use a mild heal along with the antimicrobial enzyme, now what we get the result. Suppose that we have a lysozyme, then this lysozyme, what they do, they affected the cell wall of the microorganisms. Lysozyme affect the cell wall of the microorganisms. Now, these lysozyme and the other cell wall degrading enzyme, what they do, they weak the bacteria. Now, if we combine the heat with this lysozyme and other cell degrading enzyme, then we can easily inactivate the microorganism. And how we inactivate it? By destroying it. By destroying it, by killing it. Now the next is your low pH or organic acid plus enzyme. If we decrease the pH of the food product, or if we use the organic acid along with the antimicrobial enzyme, then what is the effect of the shelf life of the food product? Okay, now we all know that the enzyme are used to break down the cell wall of the microorganisms. Okay, and making cell wall more susceptible to acid stress. Now when a food product have a more acidic value, then in that case it is very easy to destroy the microbial cell wall, because at that time the microorganism have a suspect to a acid stress, and at that time when our enzyme are attacked, then it can easily break down that cell wall of the microorganism and preserve the shelf life of the food product, which is very important. Natural antimicrobial like essential oil plus the enzyme. So enzyme weaken the structural barrier. Essential oil then penetrate the cell more effectively. Now suppose that there is a structure barrier present into the food product present near to the that microorganisms. Now these enzymes, what they do, they weak that barrier. Okay, then when we use a essential oil along with the enzyme, then when the barrier are weakened, then at the same time essential oil can easily penetrate the cell and destroy the cell wall, and can penetrate inside the cell of the microorganism and kill that microorganism, and that's how the food are preserved. Refrigeration, chilling plus enzyme. Enzyme act at low temperature, targeting residual spoilage microbes. Now enzyme can more effectively at a low temperature, means that if we decrease the temperature of the food product, then after that if we use that antimicrobial, then it can affect more as compared to the high temperature. Okay, at low temperature these enzyme target the residual of the spoilage microorganism which are present into the food product. So it can very easy for the enzyme to eliminate the microorganism, or you can say that the residual of the spoilage microorganism. Enzyme coating on packaging or active packaging. We use a enzyme to coat the packaging material in which we store the food product. So these enzyme can be immobilized on packaging surfaces or micro encapsulated into the film. Modified atmosphere packaging. Now what we have to do, we all know that this modified atmosphere packaging, that is also known as a MAP, it slowed down the growth of the aerobic bacteria. Aerobic bacteria means that it required oxygen for the growth and development. Now, these MAP, which is known as a modified atmospheric packaging, means that we modify the inside the environment of the food product which we packed. Modified the okay. Now, these modified environment slow down the growth of the aerobic bacteria because there is no availability of the oxygen. So at that time when we use enzyme, so it can easily target the only anaerobic species, mean that when the aerobic bacteria is not present or the growth is slowed down, then only the anaerobic species are present. Now anaerobic species which does not require the oxygen, so these enzyme can easily target it or the anaerobic species. Bacteriophages plus enzyme. So phages infect, and that at that time enzyme break the biofilm. When the macrophages infect the microorganism, then we use an enzyme which break down the biofilm and cell wall, and then regrowth is prevented. Combination matrix like herder technology. So each herder attack a different microbial target. Combined effect prevent the adaptation and resistance. Synergistic effect like anti enzymes plus antibiotic or that preservative, it give a multi-hurdle approach and it prevent the resistant microorganisms. Stability and activity factors like pH, temperature, iconic strength, influence activity, stabilizer, and encapsulation, they all use to improve the shelf life of the food product. Production of the antimicrobial enzyme. So it can be produced by the recombination technology, fermentation, then purification. First of all, we use what? Which technology? We use a recombinant technology in which we use a fermentation method. Then after fermentation, we purify the enzyme, and by purification of the enzyme, we get what? We get the antimicrobial enzymes. Regulatory accept like GRS status in a food, FDA, EFSA approval required. Advantages of the antimicrobial enzymes. So they are present in natural, eco-friendly, highly specific, effective at low concentration. So there is advantage like it is present in a natural way. It is eco-friendly means that it cannot affect the environment. Highly specific means that suppose that if we use a particular enzyme, then they only kill the particular type of the microorganism, and it can be even effective at very low concentration. Limitation of the antimicrobial enzyme. Narrow spectrum, sensitive to the condition, production cost. Recent advances like engineered enzyme with broader activity, fusion enzyme, nanotechnology based delivery. Case study like lysozyme in the production of cheese. It can prevent the late browning defect, extend the shelf life, and improve the safety of the cheese. Endolysin in use in medicine. It is a target antibiotic resistant bacteria, bacteriophage derived enzyme. This endolysin are what? They are the bacteriophage derived enzyme. Industrial example like DNAs in cleaning contact lenses, enzyme based disinfectant, packaging coating. Future prospect like personalized enzyme therapies, smart packaging integration, and engineered crop. Summary: antimicrobial enzymes are powerful, natural alternative to that chemical preservative combined with the other method of the food preservation, and they deliver superior safety and shelf life. Conclusion: enzyme offer a eco-friendly targeted microbial control. Means that if we use an enzyme, then they are what? They are a eco-friendly. Integration into the multiple hurdle is the future of that preservation and the therapy. So this is all about the antimicrobial enzyme. I hope you will understand, but if you have any query related to this, you are free to ask me in a comment box, or for a further detail you can follow the ITLS Academy on different platform, that is Instagram, Facebook, Twitter, YouTube, WhatsApp, and LinkedIn. And subscribe our channel ITLS Academy on YouTube and get our app from a Google Play. You can also ask your query to the helpline number that are already mentioned in your slide. That is 7080833450, and visit our website, that is www.itlsacademy.com. Thank you for watching. Please like, share, and subscribe our channel.